maybe we can we can start. So yes, it's my pleasure to introduce Eric. I have to look at your last name, Vennerson. Eric
Okay.
Vennerson, who is a researcher at KI, and he developed in 2024, I think you said, that you published a deconvolution software called deconvolve. But even though we're not going to use it today, this is there in the background if you want to check it out. And he's going to told us today about 2D and 3D deconvolution, a short lecture, and then we'll have some hands-on things. And pay attention because you will have to do the same thing as one of the portfolio assignments is that you will have to deconvolve your images. Please, Eric, thank you.
Yeah, thank you for the threats. Right, so it's the third time I talk about this, and unfortunately, it will be very sketchy because the time is brief. Just to mention, I work in the Bianco Cruzzetto Labs, which is hosted both at KI but also at Yuma Technopole in Italy. And we do a lot of things from fish design to high C. Uh, but one of our core techniques has always been wide field microscopy, and currently we use deconvolution for almost all pipelines. So, and yeah, as Sylvie said, we also have our own deconvolution software. So, back on track. What is deconvolution? And most likely, I guess you have seen what it is. You've seen some pretty examples. But after this lecture, I hope that you will appreciate it as something that is not just a prettification filter, but something more. I will try to give you a conceptual idea about what it is and point to some possible issues or problems with the technique. So I thought we can start with a thought experiment. Imagine you have prepared a microscopy slide and you put a few tiny fluorophores on it. And if you would magnify it a lot, you would see your small green fluorophores there. But what happens when we put it under the microscope? Well, one thing is that the sensor has pixels of finite size. So even if there are small details, you would not see them. So instead of seeing the tiny spots, this is more what you would see. And already that's disappointing, but as you very well know from the course, at least, there's also this actor, the point spread function, that will blur the data, which is something we can't avoid really, because it's a consequence of the physics of light. So a more realistic rendering of what you would see in the scope is this. If we now disregard noise and all the other complications. So here is actually the core of deconvolution. We would like to go from images like this back to at least the pixely version of the image, and maybe with some other techniques for spot fitting and so on, you could even find the subpixel locations of fluorophores. So is this possible? How could we go reverse this procedure to get back to the sharp pixel map? Yeah, as you know, of course, it's doable, and surprisingly enough, it's doable even by hand. And there's a game from the 90s called Minesweeper, which is actually doing exactly this. It's manual deconvolution. So instead of the fluorophores, you would look for the mines. And in this case, they have something that looks like the point spread function, so that around each mine there is something that you can detect, and it adds up linearly. So if there are many mines close by, the intensities adds up. And in some cases, you can solve these, figure out where all the mines are, and sometimes you explode them. and it goes wrong. I'd really recommend you to play this. I'm sure there are online versions if you don't have it on your computer. It's very pedagogical. So let's assume from now on there is a method, and this method probably uses the Fourier transform, which you will hear about later in the afternoon. But we just assume there is a method, a good method to reverse this blur. So why would you do it besides getting pretty pictures for your publications? I think there is one major reason, and I will try to illustrate it for you. Again, it's not real data from a microscope. Here is a simulation. Like before, I would start by putting some fluorophores in now a 3D image. And in this case, there are a lot of signals here to the right, and there's one signal which is twice as bright as any other signal in the image. And since you don't see it really, I can adjust the contrast ratio so that you only see this bright spot. But in a microscope, there's the point spread function. So what would you see if you could create this sample and put it on the microscope? It will look like this. It's not obvious that the encircled point is twice as bright as the other ones. And actually, if you adjust the contrast here, you will see that it's actually darker than the other ones, which means that you can't trust the intensities in the images unless you do deconvolution in wide field microscopy. For other super resolution techniques, the situation is better, of course. So the main reason here is that you want to be able to detect the bright spots, and if you want to know which are the brightest, you need to do something at least. And a real-world example here, a little pixely, but nevertheless, on the left, there is wide field image, which is raw, and it's a maximum intensity projection. And to the right, it's the same, but after deconvolution. If we would look at the brightest spots in the raw image, and we encircle the first 1000 local maxima, it will be those here. But rhetoric question, are those the strongest emitters? Probably not. And if we do the same, but we look for the first 1000 local maxima in the deconvolved image and put it back on the original, we see that it's a completely different set of spots. And I would argue that the spots selected on the right are the correct ones. Okay, so assuming that you want to use deconvolution, how do you do it? And I'll do the quick version. You need a license to do that, and to get that license, you need to know these four things. You need to start with high quality images, but that's also the topic of this course, mostly, I guess. So you're on the safe side there. The other thing is you have to know the point spread function. I'll say a little more about that. You need to get familiar with the algorithm or software that you use. And you also have to be critical to the results because there can be some unpleasant surprises. Right. So if we start here, you have to get the point spread function model. So there are three major ways to do it. The simplest and maybe the most attractive option is to use a theoretical one. And there you will find, for example, in the PSF generator that you will use in the workshop, where you can choose between different models. Some are simple with just a few parameters, and often they're too simple. The second option is that you measure the point spread function. How do you do that? Well, you would prepare beads, and you would image beads, small beads, and you will see how the light looks around the beads. And more advanced, you could use a parametric model and try to fit it to the measurements. Um, I'm not aware that it's used in practice, but it's been demonstrated in a few papers that it's a very good idea. And to measure the point-per function, there are also automated software that helps you to do that in some deconvolution tools. So that's about the point spread function. Um, yeah, and the reason you have to do this is that each system is unique and there are a lot of aberrations. Even if you take the same objective with the same name from the same manufacturer, they will have individual variations. And it also depends on whatever immersion media you have, the temperature of it depends on the cover slip. Did you pick the right one? And so on. And the design case is, of course, that you look just below the cover slip. So if you want to do deeper things, it might also vary. And finally, of course, the sample itself is optically active and alters the point spread function. And to the right here is a cross-section of an actual point spread function or an image of I think it's RNA fish signal, which is far from symmetrical. And if the point spread function looks like that, you might need to work a little on the PSF model to get good deconvolution results. Then there's this question about which software method to use. I would say most methods they are linear in the sense that they assume that the image is the sum of the point spread function of different places plus noise. And in this category, I would put the Richardson Lucy, which is the grandparent of most high-performing methods. And it's optimal if only Poissonian noise is included. So it doesn't really count for detector noise, and of course, for good results, you have to have a good PSF model. There are also example-based or machine learning methods that could be used. I'm not convinced that it's the right thing, but in any case, they could work very neatly if you have a lot of training data. So you need to have non-deconvolved and deconvolved data as input, or non-deconvolved plus super resolution. So it could be a viable path, but it's somewhat longer. So, I mean, I should probably give a few examples in case you haven't seen deconvolution in action. I will show mostly good ones. Let's start here. This is a sample with RNA fish probes. I think it's from my lab. Before deconvolution and after deconvolution. And here I intentionally picked a bad one because you see this ring here. And what is that? That's actually an artifact. It's because the point spread function, the model we use doesn't really match what's in the image. Here's another example. I don't remember anymore what this is. Maybe, it's some small structures nevertheless. And I took a small zoom out here. And this is what it looked before deconvolution, still wide field. And after deconvolution, they're actually small structure that start to appear, which actually makes sense. You see they should be there, more or less. Right. And of course, it can be applied to really huge images as well. Like this one is 100 and almost, it's a few megapixels. So original deconvolved. When you zoom in, you see something which is sharper, at least. What about confocal? So what's different between confocal and wide field? Well, the point spread function is more focused, it's smaller. So what would you expect? Well, you would expect smaller changes. It should be less dramatic, and it typically is. And possibly here it's even a little too many iterations because it starts to look a little spotty. But it's hard to know without any super resolution reference image. And of course, it can also be applied to STED images. This is one from Hans Blom that I guess you met before today. And here you can more or less see that each signal is a small cloud. So with some model, it's possible to get even sharper spots from that. So my very short take-home message before we workshop is that deconvolution is great, especially when the points per function is large, and that would be wide field images. And the main reason is that it provides or gives you pixel values that better corresponds to emission intensities. On the other side, it can be quite annoying because there are potential artifacts, and there are many options and parameters typically, or they're hidden for you, but they're still there. And it also used to take a long time, but nowadays it's not that big issue anymore because it's quite quick on modern hardware. And yeah, I guess we switch link from here and we
Right,
go to the
yes.
workshop.
So thank you. Do we have maybe just a little question if there is any question
Of
before
course,
we
we
go
can
to
have,
the
or we can save it for
workshop?
later.
Yeah. Do you guys have any immediate questions that you want to ask before we go to the workshop?
I mean, maybe I can answer later as well, but when you listed the different deconvolution methods, you only had one method. And I've used the Huygens software, and then you have, I don't know if it's 10 versions or at least five different versions. Will we go into in any way like how to choose the right one?
No, not here, not today. But yeah, you're right. There are many options, and although many of them share kind of similar grounds, there are many options, mostly to incorporate a priori knowledge about the homogeneity of the samples, for example, and to handle different noise sources.
So are they like more or less based on the Richardson-Lucy and then some tweaking, or is this the way it is?
Uh, Most are, but they don't have to.
Okay.
Uh,
And do you have some sort of like recommendation on how to choose one versus another?
Uh, I would start with Richardson Lucy or some accelerated version. So there are later developments that brings down the number of iterations that has to be used to get the same result. I would start there, and unless I had there are specific reasons not to use it, I would stick there. But that should be the yeah the vanilla choice. That's where you would start.
Thank you.
Okay, well, if there are no more questions, let's jump to the other Zoom then.